Okay, everyone's going to be strolling in, but I want to get started so we don't lose any more time. So our next speaker is very, very exciting to have here. As many of you know, and if not all of you know, Dr. Art Beaudet, who is coming here today with really great honor for us. For those of you who don't know, Dr. Beaudet is the one who actually identified the gene UBE3A as the cause of Angelman syndrome. So he called himself the godfather of Angelman syndrome. I think he's the father of Angelman syndrome. So I think we're really, really excited to have him here and have him talking to us. He's one of the people that really believes in our children, believes in the science, knows the science probably better than anybody in the world, and wants to see all of these therapeutics come to fruition for our kids. So it's really an honor to have him here. To give him a bio is very difficult because he apparently has about 4 or 5 different professor positions. And I'm gonna do my best here, but he is the Henry and Emma Meyer Chair and Professor of Molecular and Human Genetics at Baylor College of Medicine, a professor in the program of Integrative Molecular and Biomedical Sciences, a professor of Molecular and Cellular Biology, a professor of Pediatrics, and the Strategic Advisory Committee member of Baylor College of Genetics. Not only did he identify the gene, but he's been working his entire career in understanding the gene and the epigenetics behind the gene. So he has his main focus in Angelman syndrome and Prader-Willi, as well as the entire genome. But this is something that means a lot to him and obviously means the world to us. So it is with such a great honor for me to not only introduce Dr. Beaudet, but also to announce that we are very excited to be funding as fast this research that he's going to present to you today, and the importance of this for the future of clinical trials. So the partnership between Dr. Beaudet and FAST is really a huge honor for our community. So with all of that, Dr. Beaudet, please come up.
Either one is fine. This is easier, I think, for you. Okay, well, thank you very much. This is really my first major involvement with FAST, and I sort of refer to myself as a grandfather, not a godfather. And for those of us who were coming to the meetings when Harry Angelman was here to assume the senior role, I feel like somehow I'm the one who goes back the furthest as the time goes by. So let's see. This?
Yeah, so this is forward is green, back is red.
Okay. All right, well, I think everyone here is already thoroughly familiar with Angelman syndrome, but the first point I would like to make is that it's a particularly favorable disorder for an attempt at a pretty definitive treatment. And there's a number of very favorable circumstances about that. To begin with, the children are relatively normal at birth. They may have some problems with poor feeding initially, but if you had the average neurologist or pediatrician examine a child at birth, they would pass quite normally. And if you do imaging of the brain structure looks quite normal. And if you look early on at the brain microscopically, at least in mice, everything looks relatively normal. Of course, there are many severely disabling disorders in children where the brain is already damaged sort of beyond repair halfway through the pregnancy. So getting to birth in good health is an important asset and an important positive. And I'll be talking to you about perhaps ways to unsilence or activate the father's copy of the gene as an alternative strategy. And I think there's not many genes where you have this kind of option And it's got a lot of very attractive advantages, which I'll try to convey to you. Also, I'll show you some data we've published using what we call ASOs, or antisense oligonucleotides, that can activate the paternal copy of the gene in mice, at least. There's evidence that these kinds of molecules can be used in clinical trials. There's a disorder called SMA, or spinal muscular atrophy, and the company we're working with, Ionis, has an ongoing phase 3 clinical trial. So there's some precedent for giving this type of molecule to children with neurologic disorders, although the exact nature of the oligonucleotide would be disease-specific. And they also have pretty evidence that these will distribute well in the mouse brain, as I can show you, and I won't show you, but it's also the case that these can distribute reasonably well in the primate brain. And they can persist for surprisingly long amounts of time, that is maybe 4, 6, 8 weeks. The idea that you can unsilence or activate the paternal copy of the gene is very fortuitous, because we think it will behave much like it was a maternal copy, and it will have a lot of the normal regulation. That is, it'll be on to high levels in some cells and on and low levels in other cells. The gene actually makes multiple different proteins or isoforms, and hopefully the paternal copy of the gene would behave as would the maternal copy of the gene, and make the right distributions of the right isoforms in the right times and places. And sitting here listening to the gene therapy story today, as one thinks about, that approach, I was wondering whether, because when you do gene therapy, you have to worry about what cells are going to express and what cells are not, and what cells are what are you going to express, one isoform or another isoform. I was sitting here thinking maybe one could use gene therapy to activate the paternal copy of the gene rather than to put in a replacement gene. Now, you can, in the case of Angelman syndrome, there's something called an antisense, and I'll show you a little cartoon of that in a minute. So we are dealing with antisense to antisense here, which is a bit circular, but you can knock down a variety of genes in animals. Oligonucleotides are one approach. There are other approaches I've got listed on the slide here, which is too technical for this audience, I think. There's a consideration, the oligonucleotides that we've been using do not cross the blood-brain barrier, but there are some analogous kinds of molecules out there, particularly this U1AO silencing molecules that maybe could cross the blood-brain barrier. And there's a variety of approaches to various ways to activate the father's copy of the gene. Somehow, I live in an environment where Rett syndrome gets a lot of attention with Huda Zogby at our institution. And Rett syndrome seems to have been more well-known. But I thought it's interesting that the frequency of Rett syndrome is about 1 in 10,000 to 15,000 girls, or 1 in 20 to 30,000 overall, whereas Angelman's about 1 in 12 to 20,000 for both sexes. So there's similar numbers of individuals out there with Rett syndrome and Angelman syndrome. The Rett Syndrome Foundation, in their IRS report in 2014, raised $4 million, $4.4 million. The Angelman syndrome at that time had raised 1.7, and I'm not quite sure what FAST had raised at that time. But also, it's very interesting that if you go and search a database of publications, you find and you search under Rett, you get 24 NIH grants funded to study Rett syndrome. If you search under MECP2, which is the gene that's altered in Rett syndrome, you get 19. And if you search under Angelman, you get 4 grants. And if you search under UBE3A, you get 5 grants. So there's an enormous excess of federal research emphasis going into the Rett syndrome story compared to the Angelman syndrome story. I think because people started out viewing the Rett gene as a little bit more sexy in terms of what it does biologically, although I think now the Angelman gene is catching up. And I think maybe FAST can address this issue of private fundraising, but at a time when there's, 4 or 5 times as many grants funded by the federal government to study Rett as there are to study Angelman, it's I think good to be out there saying Angelman is a good candidate disease gene to be studying, and it's got very important biology associated with it. So let's see if I can use the, trying to see if I can point here so that you can see it on both screens, but apparently not. So I'll point on this screen over here, I guess. So this is an attempt to display the paternal copy with the bluish DNA strands here of the Angelman gene, and the maternal copy down here. The maternal copy is usually inactivated or deleted. And then there's this antisense RNA that's copied in this direction. So it's in the opposite direction that ordinarily the UBE3A gene would be copied. And this antisense now clearly is known to play a role in silencing the paternal copy of the gene. And what this story I'll talk about is that you can use oligonucleotides or ASOs to basically knock down this antisense and then allow for expression of the sense from the paternal copy of the gene. So this, in theory, would be an attractive strategy. Actually, a graduate student in my lab had done some studies that suggested that if you knock down the antisense, you can activate the paternal copy of the gene. So what she did was introduce kind of a stop signal for the RNA transcription or synthesis process in the antisense, and then demonstrated that the sense gene was, from the paternal copy of the gene, was activated. This is sort of, oops, sorry, pressed the wrong button here. This is sort of different parts of the brain expressing the Angelman gene in a normal situation. Here's the Angelman. This is another control, but over here, when you come with the introducing the stop signal, you can now get the paternal copy of the Angelman gene is turned on. So this was one way to do it, but this actually had to introduce a change in the mouse genome and wouldn't be a way that you could easily adapt to human studies. Fortunately, there was a person on her thesis committee who was aware of some of the oligonucleotide work and put us in touch with a company that was then called ISIS, but has, for obvious reasons, since changed its name to Ionis. So I think many of you here will have heard today or at many previous times information about the different forms of Angelman syndrome genetically, but the point here is that they all have a healthy, normal copy of the gene inherited from the father. In fact, the UPD patients have 2 paternal copies of the gene. And there might be a particularly fortuitous and additionally favorable situation for activating the paternal copy if you couldn't get complete activation, but only partial activation. And down here is an attempt to depict what's thought to be this collision process whereby UBE3A is being transcribed in this direction, the antisense is coming in this direction, and you get this collision, and neither one goes to completion. So it's believed that it's a fairly simple collision kind of process that mediates the turning off the father's copy of the gene. The father's copy of the gene is actually transcriptionally on. It starts copying, but then it runs into this block. So for the antisense oligonucleotides, they are 15 to 25 bases of single-stranded DNA that can match up with unique pieces of the genome. Their length is sufficient to guide them to a relatively unique location in the genome. So they have this specificity by virtue of the DNA pairing that takes place. And they can be designed to interact with DNA or RNA. In this case, actually, they're interacting with the RNA, which is the antisense RNA, and causing its degradation. There are various chemical modifications of these oligonucleotides, which you don't need to concern yourself about, although they can affect how long they hang around and how well they distribute in the brain and so on. So this is Lin Yan, who went on as part of after she finished her degree, she went on in postdoctoral work with Ionis, with Frank Rigo and Amanda Ward to do the studies, which are now published for a while, and I'll just show you a bit of. So the idea is that here we have this sense-antisense situation. In this cartoon, the maternal copy of the gene is depicted as inactive or deleted. Here we have the antisense coming this way. We introduce oligonucleotides, which then degrade the antisense and basically block it from going on down further. I think that that then leads in this cartoon to activation of the paternal copy of the gene here. I want to make the point that there are some important genes up here that are involved in Prader-Willi syndrome. If you were to turn them off, that would be detrimental. And so it is, again, there's a lot of favorable things going on in that you can come in with the antisense at this point where you can activate the father's copy of the gene without causing any problem with the Prader-Willi related genes which are nearby. So Ionis made a large number of oligonucleotides and sent them to us, and Lin Yan screened cultured brain cells from Angelman mice and looked at the ability to activate the paternal copy of the gene. In this case, this paternal copy had a fluorescent marker so that you could detect it easily when it was turned on. There had been previous work from Ben Philpott demonstrating that a drug called topotecan could activate the father's copy of the gene, but this drug has other toxic effects and wouldn't necessarily be ideal for long-term treatment in humans. So there were many oligos tested, and the higher they are on this scale, the better they worked, and you could just see that there's many that did that were quite active. And this is just the kind of thing you're looking at. This would be where the oligo is inactive. This is topotecan, which was a positive control. You see white spots in certain cells now start to appear. And with two of the individual ASOs, you could get activation that was somewhat in the same range as for topotecan. This is a mouse brain that's been injected with a dye in the ventricle, and so this is how you start out. You inject the oligonucleotide directly into the brain ventricle. In the clinical trials in spinal muscular atrophy, they're injecting the drug by the lumbar route as you would do with a spinal tap. I think if this therapy worked out in the long run, one might be able to install a pump in the brain which would pump the drug into the ventricles at controllable levels. So now we're looking in the mouse brain that has been treated, first of all, in this column we've got just detecting an oligonucleotide, and here there's no oligonucleotide added. This is a mouse that has the maternal copy of the gene expressing the fluorescent or detectable marker. Then we've got here the oligonucleotide in an oligonucleotide A, B, and a control oligonucleotide. And now you see that with an antibody, you can detect that the oligonucleotides are spread around in the brain. Although we inject them into the ventricle initially, and they start out in that watery space in the brain, they're, to me, incredibly well able to spread out further in the brain. So now if we look at the fluorescent marker to ask, is the father's copy of the gene turned on, we see here that this is when the fluorescent gene is on the mother's copy, so would be sort of what maximum activation would look like. Here's the control oligo, no activation, and here's oligonucleotide A and oligonucleotide B, showing that there's significant activation of the paternal copy of the gene by the oligonucleotide. And this is just other parts of the brain. This is the cerebellum where you have Purkinje cells, and what you see here is some variability in how well the gene is activated here. This is when it's the marked copy of the gene is on the mother's chromosome. You get pretty intense staining here, whereas there's none with the control oligonucleotide. And there's staining here, but it's not as intense as coming from when it's the mother's copy of the gene that's turned on. And this is just to show many other parts of the brain that the oligonucleotides are getting widely disseminated in the brain, and they are activating the paternal copy of the gene over a very wide area. Then we tried to look at how long the paternal copy of the gene would be turned on, and these are some studies with brain cortex and brain spinal cord. And what you see here is that in the red is the antisense is being turned off, and you see that this goes on for maybe up to 16 weeks, and the sense is being turned on in the green, and that likewise goes on to about 16 weeks. These are some of the Prader-Willi-related genes which are not altered by the oligonucleotide treatment. Oops. So I think the take-home message from the published work is that antisense oligonucleotides can be used to knock down the antisense UBE3A in cultured cells and in living mice. The ASOs target the antisense and can activate the paternal copy, again, both in cultured cells and in mice. And the ASOs injected into the brain ventricle spread widely throughout the brain and persist for up to 16 weeks. Phenotypic correction, that is, can you correct the behaviors, is challenging. We tried to look at that, and I think the results were less than ideal. One thing you frequently see is that the Angelman mice become obese, and if you look over in this panel, you can see that the Angelman mice are more obese than the wild-type mice, and the treated mice are intermediate in their weight. And this is a particular kind of learning paradigm, which it seems like there's some correction. Here's the mutant mouse, and here's the treated mouse. But these behavior studies, of which you've seen some today, are really quite challenging and tricky. And I think that it's going to be a continuing challenge to try to get behavioral correction in mice. I'm not so sure that we absolutely have to have spectacular behavior correction in mice as long as we know that the gene is being turned on properly. So one of the problems we were having with some of the behavior assays, so we had made mice, I guess now 20-ish years ago, and there was some question whether the old mouse mutation was somehow not as severely affected as it was in the beginning 20 years ago. So we tried to look at this to some extent, and we actually made a new mouse mutation. The first one we made back many years ago was deletion of a particular piece of the gene called exon 5. And then we made a new one which deletes a bigger piece of a bigger exon nearby. We've also made a rat mutation. So this particular exon, so this one was deleted in the old mouse, this is deleted in the new mouse. This is a rather large exon, and it has a number of places where the protein can start being synthesized, and these green positions are places where you could start making protein. So it's possible when you delete this exon that there could be some protein made off starting here and making the downstream part of the gene, which might have some of the biological activities of the gene. So we tried to look at that, and if we looked at the presence of RNA or product coming from the new Angelman mutation, we could see that for exons 5 and 6, that it was very low. Here's the new Angelman and the old Angelman. This is wild type, and so you could see that these exons that we actually knocked out were really not making a product. However, when we looked downstream, and here we're looking at exons 12 to 13, now the new Angelman mouse has still very little expression and looks like you might hope, whereas the old Angelman mouse is making product from these downstream parts of the gene. So we don't really know if the product that's made from the downstream part has any function or not, and we don't really know if it was there when we made the gene, when we made the mutation 20 years ago, or whether the mouse has done some kind of adaptation to express the mutation over time. So we've tried to look at how different are the old mice and the new mice. And they're not dramatically different. This is a marble burying test, which is, in my mind, somehow correlates with the absolute fascination that Angelman children have with water or running water. That is, it's a bizarre behavior that you don't see in much of any other circumstance. And we don't know quite what it means behaviorally. But Angelman mice, and if you put a normal mouse in a cage like this with bedding and with a series of marbles, they dig around and push the bedding up on top of the mice, on top of the marbles, and bury the marbles. So here you see the new Angelman mouse, and these are unburied marbles. So and there's two sexes here, males and females both don't bury marbles anywhere near as well as wild type. And the old Angelman mouse is also has this marble burying abnormality. And this is just a second period of testing the same thing later on. So this is probably one of the most reproducible behavioral activities. And the mice are not terribly different in that regard. There were some other behavior tests where it looked like there were some differences where I think maybe are summarized in this next slide, that both of the mice show neurobehavioral phenotypes or findings. The data suggests that the new one, this CRISPR one that we made, has a greater number of statistically positive hits that occur at earlier ages. But the reduced marble burying appears to be the most robust phenotype across both lines. So I'll turn now to a story of a humanized mouse and why this would be desirable. So the company Ionis, we started talking about what's the potential for a phase 1 clinical trial. And they were adamant that at least their view was the only way a phase 1 clinical trial could be done would be if we could use oligonucleotides that would be human-specific in the mouse and show that they worked and that they weren't toxic in a living mouse. So the idea was to take a piece of the human gene, this is the human antisense region, and cut it out and paste it into the mouse antisense region. These regions are not very similar in terms of their DNA sequence, and so the oligonucleotides you would use would now match up to a human target, and hopefully they would actually turn on the mouse paternal copy of the gene. And I've tried many ways around to try to convince Ionis that we could have other kinds of data that might allow for a phase 1 clinical trial, but they have been adamant that this is an absolute requirement in their opinion. There might be other companies with other strategies that felt differently. There's a lot of powerful technology to manipulate the genes of mice these days. So we thought, oh, this wouldn't be such a big problem, but Ionis wanted a really big piece of DNA moved over, and when you really try to put a really big piece of human DNA in a very specific location and orientation in the mouse, that turns out is still very challenging. So we had ideas of putting very small pieces, which would be like 50 base pairs, would be enough to be like one oligo target. And then we talked about things as big as 184,000 base pairs, so massively larger. So you can think about very big pieces or very small pieces. If you put them in this place, you would hope that the idea is the hope that the mouse antisense is coming along. It now just runs into and continues as human antisense, and then eventually goes back to mouse sequence. And so you wouldn't have to control it in any way. Hopefully it would just fit into the mouse antisense and be controlled by the mouse antisense mechanisms. If you put this piece of DNA somewhere else in the genome, that wouldn't be as nice because you couldn't activate the paternal copy of the mouse gene, but maybe you could at least look for knockdown of antisense, but you might have to then put in what's called a promoter to cause there to be RNA synthesis across this human fragment. So we've been playing with big pieces, small pieces, put into the target region versus put into other kinds of regions. And it's proven much more difficult than either we or Ionis thought would be the case. So the standard mechanisms, which now focus a lot on the so-called CRISPR technology, which you may hear about in the press, is very powerful technology, but it is not suitable for what we're trying to do at the moment. We have gotten a relatively large piece of DNA, 20 kb, which would be 20,000 base pairs of human DNA into the mouse chromosome, but not in the target region near the Angelman gene. And so we're trying to introduce a promoter to cause transcription across this 20 base pair, 20 kb piece. So we sort of have a humanized mouse of sorts, but it's not in the right position that we would prefer, and it's not being copied into antisense, so we need to make further modifications of that mouse. We've also introduced into the targeted desirable location in the mouse some sites which are sort of, I would describe as cutting and pasting sites where DNA can be cut and spliced back and forth. And so we've introduced these across the mouse region of interest. And now we can try to come back in a second step and introduce the desired piece of human information here using these as sort of a docking site. So in that kind of strategy, you could have a piece of DNA, which is here we would say that the green is irrelevant piece of DNA that we have to grow up this circular piece that we want to insert. And the circular piece has got a pink end and a red end just to say that we care about which end is which. And we can control that because we have two docking sites that are different from each other, and they function in such a way that pieces can go in, but they can't come back out. So we're now trying to introduce pieces into this docking site, which we have in the mouse. So we have a second step down this pathway, but still nothing that is entirely satisfactory. You can also introduce other kinds of docking sites, and this is a particular docking site system that is very powerful, and there's a company that's commercialized it, and so we've actually contracted with them to put this kind of docking site into the desired location, and the mice that have that have now been born but haven't been characterized. And so you can, in this case, you can put the docking site into the desired location near the Angelman gene, or it could be randomly located elsewhere in the genome. We actually have a mouse already that has this particular docking site in a random location in the genome, and we're looking at that for ways to introduce things there. So we have like 4 steps along the pathways of trying to get this kind of a mouse, but nothing that's ready to test oligonucleotides yet. I just put this up here to say that Ionis would like us to introduce really large DNA fragments. So these are, in this case, they're called BACs, which here's a portion of the human genome where the Angelman gene is over here, the Prader-Willi gene elements are over here, and the antisense region is here. And these are really huge amounts of DNA, and they would love to have some really big amount. They also are intrigued with the idea that maybe if you used an oligonucleotide against certain of these Prader-Willi elements, which are thought not to be essential, but there's multiple copies, that you might have an oligonucleotide that would be much more effective because it could hit 10 similar targets at once. So that's another avenue that they're wanting to explore. And this is just the company that has been, is putting in the target of, of a particular docking site we think will be helpful. So I want to make just a comment about the behavior in the mice. I think that a tiny amount of human data is going to be far more informative than mouse data as regards phenotypic correction. You know, the mouse phenotype is interesting, but it's also very challenging. And I think the best information is going to come when you treat an Angelman child early in life. Do you start to see any kind of changes? And so my view would be to advance to phase 1 trials at the earliest possible opportunity. Hopefully, we can get the humanized mouse that will satisfy Ionis, and we can screen a lot of oligos. And if that were available, Ionis, I think, believes they could move towards a phase 1 trial with reasonable speed. And I think they are increasingly convinced that this is a really attractive disease model among a variety of disorders they work on. This is a person in my lab, postdoc Ming Zhu, who's working on most of these humanized mouse things. And we've had funding from a variety of sources over the years, and now most recently from FAST. And so I'll stop at this point and take any questions if there's time. Okay.
There's probably intimidation, which is why no one feels smart enough to understand your slides to ask you any questions. And I'm not either, but I'm going to ask you a question anyway, which I think a lot of people are thinking about. So just if you can explain to the community, in talking with Ionis multiple times, we can't convince them otherwise that they need a humanized mouse, which seems to be incredibly difficult. But also in knowing that the rat, now that we do have a rat model, has a lot of homology on the antisense to the human as compared to the mouse. Meaning that for the audience that the sequence on the rat antisense is very similar to the human as compared to the sequence on the mouse, which is very dissimilar to the human. Would you, do you feel that using the rat would be a good
Yeah, I've tried to talk with them extensively about the pluses and minuses of the rat. And they're very adamant that they want to screen thousands of oligos, okay, across a large human region. And that it's not economically feasible to do it in the rat. And I think the rat sequence is not particularly similar in the antisense region to the human, anywhere near like, I mean, if you talk about rhesus monkeys, now that could be very much more similar. But I think both the rat and the human, and the mouse are quite dissimilar from the human in the region where there's the greatest interest in targeting.